Okay. All right. So welcome everyone. It's great to have so many attendees today. We're really excited with this series of presentation. Without further transition, I will just say a couple of words about this webinar series, why we do this, and introduce our first speaker. So yeah, NextMR, we're a company who use photo-kidnap for drug design. And we have our own bias about photo-kidnap, using it for the pharma industry, but we're really excited about all the applications that are being developed. And we thought it would be great to have, the bright mind, people who are active in the field or pushing the boundaries of this field. And yeah, we had great response from the community. We're really happy to have such great speakers today. So the first speaker that we have is Alexey Kiryutin, who is a researcher at the tomography center in Novosibirsk. And he's been doing his PhD in Novosibirsk. He's been as well with Hans Martin Witt in Berlin and in Darmstadt, working on photo-kidnap. And is also known for having developed and invented the field cycling photo-kidnap. This is particularly useful for G factors in my experience. So great to have you here. Today is going to talk to us about photo-SABRE. I don't exactly know what it means yet, but I'm really excited to discover what you have to show us, Alexey. And yeah, the floor is yours.
Yeah, thank you. Let me share my screen. Is it okay? I see.
Very well. Thank you.
So sound is fine, yeah?
Yes.
Okay. Good afternoon, colleagues. I would like to thank the organizers and Felix Torres for inviting me to speak at the seminar on light application in NMR. It's a great pleasure to present our recent results as an opening talk. This is our tomography center. It was found jointly with a Bruker in 1989, and I'm working here since was student starting from 2002. While this seminar is about NMR and its modern uses, it is worth noting how versatile the method is, from chemistry and pharmaceuticals to food science and MRI. Still, NMR has one major drawback: low sensitivity. The low sensitivity of NMR stems from the extremely weak interaction of nuclear spins with the external magnetic field. As a result, we have tiny energy splitting between levels of spin states alpha and beta, and consequently very low thermal spin polarization. At standard conditions, like a room temperature and magnetic field about 10 Tesla, we have only 0.01% of polarization. By NMR, we detect only difference between energy levels, populations. On the right-hand side, you see the case of hyperpolarization, in which all of nuclear spins have a single alpha projection. This leads to huge signal in NMR. In this case, we have an enhancement factor of 10,000. While several methods exist to enhance NMR sensitivity, including light-induced approaches, our work utilizes a parahydrogen as a source of hyperpolarization. A parahydrogen represents a spin isomer of hydrogen molecule where the proton spins are antiparallel, forming a singlet state, with a configuration makes the molecule non-magnetic and unresponsive to external magnetic field. The separation between ortho and parahydrogen is possible due to the different rotation quantum numbers achieved through simple cooling in the presence of the conversion catalyst. Here we show typical parahydrogen enrichment levels. At liquid nitrogen, we obtain about 50% of parahydrogen, while at 22 Kelvin, we achieve over 99% of parahydrogen. In our experiments, we worked with a parahydrogen enriched below or above 98%. This makes parahydrogen an inexpensive and very powerful source of spin order that can be stored in a pure form for several days without significant losses. This slide illustrates the key process of transferring the singlet spin order from parahydrogen to nitrogen nuclei in azobenzene or other substrate. The method called SABRE, single amplification by reversible exchange. It was discovered in 2009, not so long ago. The heart of a SABRE is an iridium-based complex that plays a dual role. It makes possible the reversible binding of hydrogen molecules on one side, while simultaneously coordinates substrate with a nitrogen on the other side. The chemical part of SABRE depends on many parameters, including concentrations, para-hydrogen pressure, temperature, and reaction rates. However, the physical component is equally crucial. Without proper magnetic field and scalar spin-spin couplings between the hydrides and the substrate nitrogen, the polarization transfer cannot occur. Optimal magnetic field strength varies significantly depending on the target nuclei. For efficient heteronuclear polarization transfer, for example, nitrogen, the best field is slightly below 1 microtesla, but for proton polarization, much higher field is required for which is 5 to 10 millitesla. The advantage of this method is that with continuous para-hydrogen flow by bubbling solution, for example, in the optimal magnetic field, we can achieve persistent substrate polarization. To understand the efficient polarization transfer, let's look at a simple free spin system consisting of 2 hydride protons and 1 nitrogen spin of bound substrate. When we calculate and plot the energy levels versus magnetic field, we observed that at very low field, 2 levels should theoretically cross. However, due to non-zero spin-spin coupling, these levels undergo an avoided crossing, which is called a level anti-crossing or simply lag. The position of lag depends on all spin-spin couplings in the system and gyromagnetic ratios of both hydrogen and protons, hydrogen and nitrogen. In the simplest case, where proton-proton coupling dominates, lag occurs at several hundred nanotesla, which is 100 times lower than the Earth's magnetic field. Let's consider a system initially prepared in the proton singlet state. During adiabatic magnetic field variation, we selectively populate only beta states of the X nucleus. This creates a hyperpolarized substrate that subsequently dissociates from the complex and goes to the bulk. Remarkably, when we perform experiment exactly at the lag position, we observe an analogous effect. One spin projection of the X nucleus will be populated through the same mechanism. It is important to mention the fundamental work of Konstantin Ivanov, who received at 2020 the Günther Lauken Prize for the elucidation of the SABRE mechanism shared with the Simon Duckett and Warren Warren. We remember Kostya. His brilliant career was tragically ended because of COVID. This slide presents the growing list of the molecules that can be hyperpolarized using a SABRE method. In recent years, researchers have achieved significant results with the pyruvate ion, a crucial metabolite used in cancer diagnostics via carbon-15 MRI. The SABRE method is advancing rapidly. New target molecules for SABRE are being discovered each year. Chemists develop new polarization transfer complex, including water-soluble iridium-based catalysts. And we now turn to the focus of our study, azobenzene. This remarkable molecular photoswitch exists in a thermodynamically stable trans isomer that undergoes reversible light-induced transformation. UV lights converts trans to cis isomer, but visible light or thermal relaxation drives cis back to trans isomer. On this slide, I listed several applications, including light-controlled structural modulation and even solar energy storage. Azobenzene has become the gold standard photoswitch. Anytime we need precise reversible light-driven molecular changes, we better choose azobenzene. Concerning our topic, I tell you some facts about 15N enriched azobenzene. Trans-azobenzene is planar and cannot be directly polarized via SABRE mechanism due to steric hindrance at the iridium complex, while cis azobenzene coordinates easily, yielding strong 15N polarization, which was published before. Both isomers show similar longitudinal relaxation, about 4 to 5 minutes at low fields and several seconds at high field due to strong chemical shift anisotropy relaxation mechanisms. The game-changing discovery was done by Kirill Sheberstov in our group being in Novosibirsk. He found that trans-azobenzene exhibits record long-lived spin states, almost 20 minutes lifetime under dual spin locking at high fields. Such long-lived spin states can exist only in magnetically equivalent nuclei with high symmetry in the rigid molecules. By the way, parahydrogen is the simplest case, which can live for many days. LLS in cis isomer lives only several seconds, just slightly longer than T1. This phenomenon of long-lived states, well developed by Malcolm Levitt group and his group in Southampton, it actually opened new horizons. When we realized these facts 4 years ago, our mission became clear: to hyperpolarize trans azobenzene by SABRE and lock its polarization for 20 minutes in his long-lived states. We came to the idea of photo-SABRE. Our goal was to combine all our existing capabilities while solving several critical technical problems simultaneously. First, we needed to introduce light into the magnetic field without distorting the ultra-low field environment. Even a minor disturbance would ruin the polarization transfer. Second, the system had to maintain high-pressure parahydrogen delivery up to 10 bars, while accommodating this optical setup. And crucially, we had to preserve sub-Hertz NMR resolution despite the sample shuttling rapidly between fields. I'll be honest, this was an incredibly exciting challenge. The difficulty of getting everything to work together: light illumination, parahydrogen bubbling, sample shuttling, field cycling in the magnetic field, and NMR detection with long-lived state option. To perform SABRE, a measurement in arbitrary magnetic fields, we already had a setup based on moving the sample along the axis of the cryomagnet, which I constructed in 2016. This setup allows us to vary magnetic field from 2 millitesla to 9 Tesla by positioning the sample. At the top of the cryomagnet, a magnetic shield is installed with internal electromagnets, enabling the variation of magnetic field from 10 nanotesla to few millitesla. Thus, we cover 9 orders of magnitude in the magnetic field strengths. A special adapter for screw cap NMR tube equipped with the Teflon tubes allows us to bubble the solution under pressures up to 10 bars. The gas system enables sequential delivery of 3 different gases. From the NMR side, we can use standard TXI and BBO probes while keeping high resolution and reproducibility. The setup is highly robust. Since it starts, it has performed over 2 million sample movements. The video clip demonstrates real-time sample movement, which is sufficiently for the majority of experiments. The whole setup is controlled by the PC computer directly from top spin pulse program. By the way, the similar setup I have built in Darmstadt in Gerd Bunkowski group in Germany, and copy of it was constructed in University of Southampton in a Malcolm Levitt group. To irradiate the sample inside the magnetic field, I initially used a simple LED of various wavelengths arranged around the perimeter of the guiding tube to illuminate the sample from all sides. From the perspective of illumination, this setup worked well. However, there was a problem with the magnetic field. It turned out that LED was slightly magnetic, but more important, the current flowing through them produced additional magnetic field, which significantly disturbs the ultra-low magnetic field, which was unacceptable for our experiment. So I decided to place the light source outside the magnetic field and deliver light using 2 mirrored surfaces, as it is done in the periscope. This approach required the use of small dyad lasers with a non-divergent beams. Examples of such inserts are shown on the slide. At the first stage, I used 5 lasers, but in the final setup shown here, I used 7 high-power dyad lasers with a wavelength of 450 nanometers to produce more light power. The total electrical power is 9 watts, and the optical output reaches 1.2 watts. Aluminum heat sinks you see with the small fans were used to dissipate heat from the lasers, but to cool the sample exposed to the intense light, we used air channels inside the walls of this tube. This design turned out to be very successful. It is easy to install in our existing magnetic shield and allows for highly efficient sample irradiation. I have to say that all of this became possible due to 3D printing. The updated design could be fabricated within a single day without even leaving the lab. The slide shows a series of proton NMR spectra of azobenzene recorded after continuous green light irradiation every 3 seconds. One can clearly see the gradual transformation of trans-azobenzene into the cis isomer. On the right, photoisomerization curves are shown for different concentration of azobenzene. The higher the concentration, the slower the photoisomerization proceeds, which is explained by the increased optical density. These initial experiments demonstrated the importance of delivering as much light as possible to the sample in order to be fast to convert trans to cis and cis to trans. So we applied the protocol shown on the right. We irradiated a prepared solution containing a mixture of cis and trans azobenzene and activated iridium complex in an ultra-low magnetic field while simultaneously bubbling parahydrogen through it. First, we looked at the proton NMR spectra. It turned out that without light, only the cis azobenzene is polarized, whereas under light, both isomers become polarized. We then repeated the measurement for 15N and even for carbon nuclei. In this way, we converted our dreams into reality. Continuous light irradiation simultaneously converts cis to trans and trans to cis, allowing us to polarize trans azobenzene. In other words, light activates the chemical exchange between the two isomers. The molecular photoisomerization proceeds very rapidly, so the spin polarization is not lost during the transition. To measure the magnetic field dependence of polarization transfer, we conducted a series of experiments in which the field inside the magnetic shield was varied. We found optimal magnetic fields for different types of nuclei, but most importantly, we confirmed that the field dependence profiles for cis and trans azobenzene are identical, differing only in amplitude. I won't go into details about the field dependence here, but I want to mention that for 15N nuclei, the optimal magnetic field is around half of microtesla. This table shows the maximum enhancement factors and polarization levels achieved under optimal conditions. As it is seen, the polarization on nitrogen nuclei reaches 7.5% for cis isomer and 3% for trans azobenzene, which is very good results. I'd like to point out that in such a system, achieving the highest enhancement factors requires additional experimentation. For instance, the concentration of azobenzene is a key parameter. It affects both the optical density and the exchange rate with the iridium complex. I suspect that this system can be tuned and polarized even stronger. We also performed experiments varying the irradiation and bubbling times, the so-called build-up curve. It shows how the polarization on trans azobenzene slow increases all the time. In this case, the rate of photoisomerization on the entire sample volume plays a crucial role, and also mixing of the sample plays a crucial role. We also carried out a relaxation experiment in the dark. This showed that in ultra-low magnetic fields and in the Earth's field, 50 microtesla, cis azobenzene relaxes very rapidly in the presence of the iridium complex, while trans azobenzene exhibits a T1 relaxation time about 100 or 200 seconds. The SABRE experiments with azobenzene were highly reproducible, and the sample showed virtually no degradation, which allowed us to perform relaxation measurement in a magnetic field range from 1 millitesla to 9 tesla. The protocol of this experiment is shown on this slide. The so-called relaxation dispersion, or T1 dependence on a magnetic field, for the two azobenzene isomers is shown on the slide. When compared to the data obtained by Kirill Sheberstov for the azobenzene, thermal azobenzene in acetonitrile, we see the T1 behavior of the trans isomer remains unchanged. In contrast, the cis isomer shows significantly faster relaxation in magnetic field below 100 millitesla. This can be explained by chemical binding to the bulky iridium complex, where the relaxation time is evidently shorter. The sharp drop in the strong magnetic fields is attributed to the chemical shift anisotropy mechanism, and the quadratic increase in relaxation rate with the field strength has been observed. The final part of my talk concerns long-lived spin states. As I mentioned earlier, LLS can exist in a system with high symmetry, with a spin 1/2 pairs, or in system of A prime, M prime kind. There are pulse sequences designed for the detection of long-lived spin states. Two examples are shown here. For example, S2M pulse sequence and slick spin lock-induced crossing. We tested both approach and found that the S2M sequence works better for the case where the chemical shift is slightly changes due to, for example, temperature variation. The spectra show how the 15N signal of polarized trans azobenzene looks like when we detect with a slick pulse sequence. A doublet is observed, which is expected compared to the conventional magnetization-based detection. It was interesting to investigate the magnetic field at which photo-SABRE is most effectively in populating long-lived states. It turned out that zero magnetic field is the most efficient. The small dip here in the center is an artifact of the experimental setup near zero field. Residual transverse field component of about 30 to 50 nanotesla remains, which can significantly affect measurement in this range. The last slide shows the spectra of trans azobenzene and the relaxation kinetics in logarithmic scale for magnetization at 9 Tesla, at 0.1 Tesla, as well as the decay kinetics of long-lived spin order in high field under double RF spin lock. It is clear seen that the LLS signal persists even after 17 minutes, while the magnetization has completely decayed. On the right side, proton NMR spectra of the long-lived spin order of the orthoprotons are shown. Fitting the data allowed us to determine the relaxation times of the combined multinuclear 1H and 15N long-lived spin order, which is 1,200 and here 1,000, almost 500 seconds, corresponding to 20 and 25 minutes, respectively. So in conclusions, for the first time, we have successfully hyperpolarized trans azobenzene using photo-SABRE method under light radiation, achieving a 15N signal enhancement more than 9,000, which is 3%. Polarization is transferred from the cis isomer, which is directly polarized via SABRE at ultra-low magnetic fields. The long-lived spin order of trans azobenzene populated directly during SABRE at near zero field exhibits a remarkable lifetime of 25 minutes, far exceeding its T1 relaxation time at high field. This multinuclear spin order can be detected via both 15N and proton NMR. This work bridges photochemistry and hyperpolarization, enabling new application for light-responsive system in MRI and NMR. So results are published in this paper. And the last slide, here is our team with whom I was working in Novosibirsk. I'm thankful to all of them, but here I highlighted who are involved in this work. So thank you for your kind attention.
Thank you, Alexey. Thank you so much. It was an amazing presentation. Beautiful science.
Yeah.
And yeah, I think do we have any questions already in the Q&A in the chat? Yes, I see a question from.
How to. Okay.
I find the
now
questions. Don't worry. So we have a question from Ron Lawler, Professor Ron Lawler, who asks, have you considered using substituted azobenzenes?
Actually not. So you see, we used a doubly labeled 15N azobenzene, which we got from Herbert Zimmermann, and we actually we tried to use other Azobenzene water-soluble, but they are not 15 labeled. So we tried only at natural abundance, but with natural abundance, first of all, signal is tiny, but also we found out that N14 causes relaxation very fast. So actually we tried, but we would like to have 15N labeled substituted Azobenzene. It's interesting topic, yeah.
This makes sense. We have a question from Sadam Adebayo. Thank you for the good work. My question is, does this signal enhancement help in the acquisition of 2D NMR spectra?
Oh, yeah, sure. It's possible to, I think I have even done a 2D experiment with the hyperpolarized azobenzene. I think it was kind of noisy or XE experiments. Yeah, sure. Within single scan, you can acquire one transient, and then the important thing that it should be reproducible, but our setup actually quite reproducible, and every transient could be started with the same, almost with the same amount of polarization. Yeah, it's possible.
Maybe I have a follow-up question for you, because one thing that I remember from 2D's hyperpolarization is often we can use gradients, for instance, to measure 2D within one scan. And because one challenge is often to recycle the hyperpolarization from scan to scan. So we tend to either do that or do small angle excitation, so that at least we can record 2D. We can keep some polarization in the Z axis. And in photo-kidnap, for instance, you can't simply recycle polarization, the limit of bleaching by pulsing again light and light. Is that possible with the field cycling and the para-hydrogen bubbling? Would you be able to do that with your system, to just bring the sample up
it's
and
possible.
down?
So the SABRE is very nice method actually. So it's quite reversible. So you can produce your hyperpolarized signal just in 1 day or even 2 days, just bubbling and measure bubbling. What you need to take care, about evaporation of the solvent. But this, if you bubble a lot with a dry hydrogen, you can evaporate your solution or solvents. But this can be solved by pre-moisturing, so to say. So, in gas phase, you can add vapors of your solvents, and then you can bubble for several days. And for some system, it's reproducible. For some, of course, not. But it's, as I remember, since I did Kidnap is really fast degrading system. Usually you cannot do 1 hour illumination. It will deplete it.
No.
Yeah.
Absolutely. I will finish maybe with a last question, which is a follow-up question from Sadam Adebayo, which is there any constraint of temperature for maintaining the long-lived state?
Temperature. Well, I think since it's in a liquid state and you don't have any kind of phase transition, meaning crystallization, it should be fine. So we actually didn't change so much temperature, did not vary. But I think if it's in a liquid and in the same, let's say, phase, I don't think that temperature will change a lot. Yeah.
Excellent. Thanks a lot. Yeah, I think we are in time to move to the next speaker. Thanks a lot, Alexey. Again, very interesting. I can see a lot of potential applications going down to MRI eventually one day. So that's yeah, that's where it starts. Really, really nice. Thanks a lot.
Thank you.
And with that, I will start to introduce our next speaker, who is Lars Kuhn, Dr. Lars Kuhn, who is now a researcher in University of Freiburg in Germany, and he's been studying chemical science in Bonn, Oxford, Paris, and Strasbourg. So you've been to my home country, France. And he's as well been working with hydrogen polarization, but with FIP, if I'm not mistaken, not with SABRE. And yeah, today he's going to talk to us about how we could apply photochemically induced dynamic nuclear polarization to metabolomics. And that's a very hot, metabolomics by NMR is a very hot field. Photokidnap is a hot field. So together, let's see what it gives. And really excited to hear this talk as well. Lars, the floor is yours.
Okay. Thank you very much indeed, Felix. I hope it all works. Yeah, thank you very much indeed for having us today. I think it's a great opportunity, and I think it's a great idea to having set up this series of webinars. Yeah, I'd like to talk about a project that we have been working on now for quite a while, and yeah, which has come to completion roughly about a year ago. And yeah, as the title suggests, we've tried to apply PHOTO-KIDNAP to the analysis of complex mixtures of biological origin. Just wanted to start by saying hello from Freiburg. Just for those of you who don't know where this is located, Freiburg is a small university town located in the deep southwest of Germany. We're bordering both France and Switzerland. And at the same time, we're also stretching the western slopes of the Black Forest. This is a low mountain range in the southwest of Germany you might have heard of. So just for you to know where we are. Right, yeah, as we all know, NMR is great. I suppose this is why we all like or even love it so much, and there must be a reason for this. So NMR has a lot of upsides or advantages compared to many other analytical techniques, and that's also one of the reasons why it's being used in the field of metabolomics. I just wanted to point out a few of those reasons. I mean, as we all know, NMR is normally a non-selective technique. That is, everything that's in your NMR tube also shows up in the spectrum, at least as long as the concentration of the analyte of interest is above the detection limit of the method. Another advantage is, and that's very important in the area of metabolomics, it's inherently quantitative, so that's very good and very important. The results that you obtain from an NMR measurement are normally highly reproducible, so you can repeat the experiment over and over again, and as long as you do not change the experimental conditions or the composition of your sample, you should normally always end up with the same result. Very importantly, NMR is a non-destructive technique. This is the big advantage as compared to mass spectrometry, for example. That meaning once you've measured your sample, you can take it out of the magnet and you can just subject it to any other kind of analysis. So this is just awesome. Normally, at least, NMR sample preparation is rather straightforward. Yeah, I would call it even minimal in most cases. And yeah, of course, the big advantage is if you want to know more about the structure of a molecule, NMR supplies you with a huge amount of structural data or information in order to analyze the structure or any other characteristics of your molecules of interest. As every method, NMR also has a few downsides. I mean, we've all heard about this many times before. It is a very insensitive method, and this is what we, in one way or the other, I think, all try to mitigate. And the other problem, if it comes to the analysis of complex mixtures, of course, is that the NMR spectrum becomes rather crowded, and this really makes the assignment and the analysis of the data in many cases relatively difficult. That's it. And just to give you an example, this is, for example, the proton NMR spectrum of a sample of the normal human urine. This is a sample that you very often deal with in metabolomics, yeah, because it is full of clinical or medically relevant information, of course. And as you can see here, the NMR spectra of these mixtures are hugely crowded, especially in this case in the aliphatic part of the spectrum, but also in the aromatic part. And it's not only that we have many signals around in the spectrum, but also that the concentration range of the analytes that we're dealing with is very different. I mean, you have some analytes which are rather highly concentrated, and at the same time, you have other analytes which occur in a very low concentration. And this, of course, makes the discovery or the assignment of those signals even more challenging. And to address the sensitivity issue of NMR, also in the context of biofluids, hyperpolarization methods have already been applied to tackle this problem and to simplify or to make the analysis of those measurements easier. There's of course, the field of dissolution DNP, which is a very popular method these days, and it has also been applied to the analysis of complex biological mixtures. As far as I can see, the main player here is Patrick Giroudot from the University of Nantes in France, together with his co-workers. So they have applied dissolution DNP for the analysis of metabolomic samples, and they have also run entire metabolomic studies based on this. So they have been extremely successful in the recent past. As far as the parahydrogen world, I should call it, is concerned, I was able to identify mainly two groups that have been working on this. On the one hand, there's the group of Marco Tessari from the University of Nijmegen in the Netherlands, and there's also another group working in, I think it's Estonia, if I'm not mistaken, who have also contributed significantly to applying parahydrogen-based methods to the analysis of biofluids. So when we started this project, we were basically asking ourselves, dissolution DNP has been applied, parahydrogen has been applied, and if you come from a photokidnap background, it's kind of obvious that you ask yourself, has anyone in the photokidnap area or field ever applied this method for the analysis of complex mixtures? And we screened the literature, and well, we didn't really find too much, to be honest. And this is probably the main incentive for us, or was the main incentive for us to give it a shot and to just try it out and see what happens. I mean, most of you, I guess, are familiar with the photokidnap method. Unfortunately, I do not have the time today to go into the intricacies of it. This will probably take up too much time today. But for those of you who are not too familiar with this method, let me just quickly tell you that as of relatively recently, photokidnap was a relative selective NMR hyperpolarization method, at least as far as small biologically relevant molecules were concerned. It was basically only a handful of molecules that were known to be amenable to the photokidnap effect. I will tell a bit more about that just in a second. As far as the origin of the hyperpolarization is concerned, there's this radical pair mechanism, which is mainly responsible, at least in the solution, for the generation of the hyperpolarization. You normally start off with a photosensitizer or a dye molecule. In most cases, this is a flavin derivative. I mean, there are other dyes around these days, but we've been using the, I would say, old-fashioned one, flavin mononucleotide, to basically start this photocycle, which eventually generates hyperpolarization in the substrate of interest. And what it all boils down to is basically, if you consider a pool of protons in a static magnetic field, you either overpopulate the alpha state of this proton, and this will lead to enhanced absorption in the resulting hyperpolarization spectrum. Alternatively, you can also overpopulate the beta state, which will then give rise to emissive signals, and you can have both of them in the same spectrum at the same time. So it's a bit unique as far as hyperpolarization methods are concerned. And until very recently, the dogma of biological photokidnap was that it's extremely selective, because there's one requirement, and that is that the molecule that you want to hyperpolarize should have a low ionization energy in order for this spin correlated radical pair to be formed, which eventually gives rise to the hyperpolarization in the NMR spectrum. And well, we have to thank our host and his co-workers, who have been able in the very recent past to significantly widen the chemical space which is amenable to the photokidnap effect. I think, Felix, correct me if I'm wrong, but I think you've probably now established libraries comprising a few hundred, if not even more, substrates that are at least potentially polarizable using the photo-KIDNAP effect. So this is really great, and I think it gave a real push to the entire field, and it was also one incentive for us to work on this. Right, just briefly a bit of our illumination setup. Nothing really fancy here, to be honest. I mean, we're using a very established way of illuminating our sample. It's a design that was published, if I'm not wrong, in the early or the mid-80s by Larry Berliner and co-workers, and it basically requires the use of this coaxial glass insert, which is normally used for chemical shift referencing. But it's good enough to also attach an optical fiber. So in our case, our waveguide comes from above into the bore of the NMR magnet and dips right into the sample solution, as you can see here in the picture. And this is our way of illumination. I know there are many, or there are much better ways of illuminating your sample available these days, but we had this setup available. That was one of the main reasons why we wanted to use it. At the far end, this fiber is connected, of course, to the light source, which in our case is a CW diode laser emitting at a wavelength of 445 nanometers, with which, of course, we try to hit one of the absorption maxima of our photosensitizer, which is, of course, necessary to get a good triplet yield upon light excitation at a relatively moderate laser power of 400 milliwatt maximum. Normally what comes out at the end of the fiber is quite a bit less. Okay, so as I mentioned before, KIDNAP is quite selective, and this is one of the reasons why we thought it might help simplifying over-complex NMR spectra of biofluids or biological mixtures. And in order to test whether this also worked in our case, and also in order to test our NMR setup that we had available, we basically started off with a test experiment. We basically prepared a mixture, an aqueous mixture of the 20 amino acids that occur naturally. And as you can see here in the proton spectrum, what we expected to see was a very crowded spectrum, especially in the aliphatic part. That's, of course, because most of those amino acids have aliphatic side chains, but also there are a few aromatic ones around. So even the aromatic region of the NMR spectrum is quite a bit congested. We then subjected this sample to, well, a photo-KIDNAP measurement. I mean, we added the photosensitizer in a relatively low concentration, I think around 0.2 millimolar. That is, this is the concentration that we normally use for these experiments, and this is what we ended up with. Obviously the spectra is hugely simplified. We only see a few resonances out of the many that were present in the thermal spectrum, and of course, I know all our critics can argue, yeah, but you lose a lot of information here. And that's true. And that's also I wanted to make clear at the very beginning. We are not trying to compete here with any other method. We think this might be eventually a complementary way of trying to overcome the overcomplexity of these, the rather difficult NMR spectra, or let's see, at least difficult to analyze. So eventually, out of all these 20 amino acids, we knew that 4 of them should be in principle polarizable with this method, and this is also what we saw. So at least we knew that our setup was working in our case. The sequence that we used, I mean, we didn't apply difference spectroscopy. For those of you who know that, normally in KIDNAP, you first acquire a dark spectrum without laser excitation, and then subsequently you acquire a light spectrum, and then you subtract the two from each other, just in order to get the net KIDNAP effect. In our case, we use the pulse sequence, which does that internally. So this is a pure 1D photo-KIDNAP spectrum. Right, so this was quite successful for us, we thought, and the next step was then to change the matrix of our sample and go for a sample of human urine to begin with as a good test sample for our purposes. And oops, sorry. What we then did was we spiked the sample with these 4 amino acids of which we knew that are potentially polarizable with the photo-KIDNAP method, and we wanted to see if this complex background had any effect on the generation of the hyperpolarization. And this is the result. Below, again, the thermal spectrum, and above you can see the hyperpolarization spectrum. And basically, again, at least qualitatively speaking, we were able to detect all those amino acids that we hoped for at the beginning. Those are the ones that we knew were photo-CIDNP active. And on top of that, marked here with an asterisk, we were also able to see some additional polarization signals, which were not stemming from those 4 amino acids. And we already had an idea that those might actually come from metabolites that are also present in the sample solution. Right, so this was quite encouraging. And yeah, eventually we then went for the full Monty and used an untreated and fully pristine sample of human urine, to which we really only added the FMN, because of course without the FMN we knew it wouldn't work. And of course we added some D2O for the NMR experiment. And this is then the next spectrum. This is then what we ended up with. Again, below in gray, you can see the thermal spectrum, quite congested, a lot of quite many signals, quite difficult to analyze. And above, the hyperpolarization spectrum. And yeah, we were lucky enough to see a few signals, especially, of course, in the aromatic part. This was not too surprising, but even in the aliphatic region of the spectrum, 2 prominent signals showed up. And when we enlarged this aromatic part, which of course was more relevant for us, we saw a few of quite a number of hyperpolarized resonances, not only those that we were expecting, like from tryptophan, for example, or from tyrosine, but also a few unknowns. And the most intense of them were then subjected to a further analysis, because of course we wanted to know which molecules are responsible for the hyperpolarization signals here that we were able to observe. I wanted to point out one thing here in this spectrum. You can see that most of the hyperpolarized signals have a counterpart in the thermal spectrum, which of course was to be expected. They were only sensitivity enhanced. But if you look here, the region of around 6.2 or 6.3 ppm, or even 6.4, we also detected a few emissive resonances who do not have a thermal counterpart. I mean, these spectra were acquired with 16 scans, if I'm not mistaken. And in the thermal spectrum, you are not able to see them, whereas in the photo-CIDNP spectrum, they show up. So there is a bit of an advantage of the sensitivity enhancement as well, even though I have to point out in these experiments, we really didn't go for maximum sensitivity. I think this is something for the future. Right, okay, so we subjected those unknown signals to a chemical shift analysis. We compared the chemical shifts with databases. We also looked at the hyperpolarization patterns, and we also did some selective 1D toxi analysis in order to deconvolute all these crowded signals that you can see here in the aromatic region of the spectrum. And this is then basically what we ended up with. Eventually, we were able to identify these 10, I think it's 10, yep, 10 metabolites that were assigned in the end unambiguously. And we then checked whether those metabolites also in one way or another have a clinical relevance. And we found aberrant physiological states that are indicative of either an up or a down regulation of those metabolites in human urine. And as such, and at least as far as the future of this method, if there is a future, who knows, is concerned, we think, this has quite a bit of potential, at least as far as targeted analysis are concerned. So for those of you who don't know about this, in targeted metabolomics, you know about the candidates you're looking for before you start the analysis, whereas in an untargeted analysis, you just look at all the metabolites available in the mixture. Right. Good. This is just another slide to explain to you how this 1D toxi analysis works. You basically irradiate just one resonance and you look for those other resonances that show up due to toxi at the same time, and this will give you some information about which of these signals belong to the same molecule and which signals stem from different molecules. So this was quite helpful during the analysis. Before I finish up, just 2 more things to mention. I mean, if you're about to establish a new method, people will always ask you, do you think it's reproducible? And what we did to demonstrate that this is, according to what we think, quite reproducible, we prepared independently 3 different samples from scratch, and we basically subjected them again to the same method. And well, as you can see here, the error that we obtained was quite small. So we think the method is relatively reproducible, or at least can be reproducible in the future. An even more important aspect of a newly established method in the field of metabolomics, of course, is the question whether it provides quantitative data. And that's maybe a bit of a downside of CW-KIDNP. CW-KIDNP will probably never be able to provide you with quantitative data because mostly you just illuminate for too long. So there's a few side reactions that also occur at the same time. And also different photo-KIDNP active molecules, they compete for triplet excited flavin. And so probably CW-KIDNP will never be able to provide quantitative data. However, for anyone who wants to work on this, maybe us, maybe others in the future, we think the way to go is to use time-resolved KIDNP. That requires a different setup. Basically, you don't work with a CW laser, but rather you work with lasers that that basically emit ultra-short pulses, ultra-short laser pulses, and they allow you to also observe the KIDNP effect, and you get rid of many of those side reactions which can obscure quantitative information. And just to give you an idea how this could work in the future, we applied a so-called standard addition method to our data. We basically looked for one analyte specifically in our biomixture. In this case, that was L-tryptophan. And what you do in this method is you basically dope your sample with different concentrations of the analyte of interest, and then you acquire the photo-KIDNP signal. In our case, Patrick Giraudoux does the same for dissolution DNP. And if you then plot the intensity that you obtain versus the concentration that you added, you get normally a straight line, at least if the method that you use is quantitative. And if you look at the y-intercept of this straight line, this will basically give you an idea about the concentration of the analyte as it was in the untreated sample. So we think in the future, this is probably the way to go if anyone wants to make this quantitative. And yeah, this is our suggestion at least. So I'm at the end of the presentation. I just want to quickly conclude and tell you that at least we've demonstrated the proof of principle here. And we were able to see those hyperpolarized resonances using a complex biofluid. We were able to hyperpolarize roughly 10, maybe even a bit more. We didn't analyze all of the unknowns metabolites, so this was quite good. And yeah, as I showed to you, I think the method is quite robust. It is minimally invasive, given that we use NMR, and KIDNP is not really such an invasive method. And as such, we think in the future it might one day it might be incorporable into standard metabolomics protocols, which of course afford high throughput screening of a huge number of samples. So in metabolomics, you don't measure 2, 10, or 20 samples, but normally, if you want to get statistically relevant results, you should measure maybe at least 100 or maybe 200 or 300. And yeah, so there's a lot of work to be done. This method is in its infancy. And we would have to work both on maximizing the sensitivity to define a class of potentially polarizable metabolic targets. And of course, very important here to establish quantifiability of the hyperpolarization data. Right, I just wanted to finish up by thanking my main collaborator, which is Miriam from the Autonomous University in Barcelona, together with her boss, Theo Perella. I'd like to thank my host in Freiburg, which is Stefan Weber, and I would like to thank my long-term collaborator, mentor, and friend, Joe Bargon, without whom I would probably not be sitting here. Last, I'd like to thank the donors of all the samples that we used. They submitted them to the University Hospital in Madrid, La Princesa. And that's it. Thank you very much for your attention.
Thank you very much, Lars. Really nice talk. I think that's quite remarkable as well that you mentioned as well guided metabolomics, where you know what you're looking for, and this doping trick is really exciting. And I see that people in the chat are as well excited. We already have 2 questions. The first one is from Marta Stefanska. Thank you for the nice talk. Did you remove oxygen from your samples and how? And do you foresee any matrix interference with biofluid that could lower the photo-kidnap enhancement?
Well, I mean, the oxygen, of course, you wonder because oxygen, of course, can quench triplet flavin. Is that the reason why you're asking? I mean, oxygen there's always two things about removing oxygen from your solution. On the one hand, of course, I mean, it's a triplet oxygen, so it's a radical in itself, so it might quench the amount of triplet flavin available in solution. But at the same time, it also prevents the regeneration of photoreduced FMN, which of course leads to photo-bleaching. So we didn't do anything of this because we didn't really try to maximize photo-kidnap sensitivity here. I think if we were to try to maximize it, we would probably look for other things. I mean, I know in the past people have been experimenting with H2O2, I mean, hydrogen peroxide. Question is, again, will it interfere with anything else in the sample? We don't know that. We haven't tried that yet. And the other question, I think, but I mean, I think one way to go is probably, I mean, Silvia Cavaniero has developed so many different ways of maximizing your kidnap sensitivity. So I think you should look into her toolbox, and you will probably find a lot of very useful things to use. I mean, you might be thinking about using this tri-enzyme mix that she's proposed, or maybe others. But then again, you will always have to think about, will it interfere with your sample components or not? It's complex. It's really complex because we're dealing with complex mixtures. And the other question was, sorry.
And do you foresee any matrix interference with biofluid that could lower the photo-kidnap enhancement?
I mean, as I said, we didn't analyze it quantitatively. Of course, we will have to do that, or someone will have to do that, because this is the bottom line of this piece of research. I'm sure the matrix has an effect. So on average, we could see that signal intensity went down. Of course, if compared with an aqueous solution, yeah, it's not the same. I mean, for the spin correlated radical pair to form, you need a solvent cage. So it's all a bit tricky around there. If you have other molecules, maybe they interfere with the formation of the solvent cage. We don't really know. I mean, in urine, you have maybe a few thousand different molecules and entities present, right? It's not only molecules as such. You also have metals, you have salt. I mean, you have all kinds of stuff. So in principle, there's a lot of different factors that could lower your kidnap. But this is something, I guess, it remains to be seen. And yeah, in principle, there's a lot of work to be done, I think. Yeah. We didn't check that, no.
All right. I think we have time for one more question. So we have another question from Covadonga Lucas Torres. Thank you very much for the wonderful work. Is it difficult to adjust the photosensitizer ratio considering how different metabolic concentrations could be in the biofluid?
I mean, normally the main limiting factor of using too much FMN, I mean, people or experts might say something else, but is the optical density, of course, of your solution. I mean, FMN strongly absorbs where we emit with our laser. So we just use the standard concentration that we've always been using, even if we were doing photo-kidnap of proteins or amino acids. So it's always been around 0.2. If you increase it further, I mean, the optical density of your solution will go up big time. And the way we illuminate, we really have to make sure that the optical density of our solution is not too high. And that's because, the way we do it is basically our light comes out of the optical fiber in a cone-shaped manner. And of course, the intensity will drop, I mean, hugely over the entire route of the coil region. We've never used smaller amounts either, because this will just lead to a smaller amount of signal enhancement. So, but this is of course another parameter you could play around with and see what's going to happen. Maybe in the case of a complex mixture, different amounts of photosensitizer can be advantageous. We don't know that yet, but at the moment, we just wanted to keep all the other parameters, especially the kidnap parameters, equal, so as to not have any further reasons to believe something is due to the matrix or not. But again, this was like a first proof of principle pilot study, if you like.
Yeah, that's really nice work. I think we have some more questions, but we have to move on. Just for a reminder for everyone, the questions that are not answered now, we can either ask them again at the end if the speakers have the time, depending the different time zones. And anyway, I will send the questions by email to the speakers, so they will have the chance to answer them, and it will be sent to the attendees by email with a small report. So like this, you don't miss on anything. So don't hesitate to ask your questions, even if we cannot cover everything, to make sure people with different time zones as well don't end up too late. Yeah, this will be answered maybe not today, but at least in the next couple of days. All right, Lars, thank you so much.
Thank you.
Really, really nice talk. I think that will be the beginning of something. I'm really excited to see what will come up after this. And we will move on to our next talk with Guzel Musabirova. And she's a postdoc in Leipzig University in the group of Professor Jörg Matisic, and she's a member of the HIPMOL, which is a German research network for hyperpolarization in molecular systems. She's been working. Oh, sorry, I lost my note. So she's it's in the Transregio by the German Research Foundation. She did a PhD in biophysics at the Kazan Federal University in Russia, and she's been working as well on the spatial structure of complexes of statin with model membrane using NMR spectroscopy with Professor Vladimir Kloshkov. And today she's going to talk about distance-dependent photo-kidnap in polyproline-based dyads, which is something that we see a lot as well in enzymes. So in biomolecular systems. So yeah, really excited to hear about your research, and the floor is yours.
Oh, thank you for this nice introduction. Actually, I find very nice idea to organize such kind of seminars, and really thank to that you give an opportunity to share the work and then get a lot of opinions. So let's start. Today I will talk about distance-dependent photo-kidnap effect in polyproline-based dyad. As already was mentioned, this work is done within the HIPMOL consortium. And before to start, I would like to go to the background and explain where the idea comes from. So, as we know, many biological systems use light for signaling and control. And one fascinating example is blue light sensors. They are found in many organisms, bacteria, plants, algae, animals, humans. And these sensors are called blue light photoreceptors. They are kind of special proteins that can detect blue light and trigger specific biological responses. And here is a flavin. It's a molecule that plays a key role in the absorbing light and initiating the photoreceptor response. And depending on the protein environment and molecular modifications, flavin is a part of the different type of photoreceptors, such as blue light using flavin. They are found in bacteria and algae, and they regulate photosynthetic chains and help organisms adapt to light conditions. Another example is light oxygen voltage domains. They are part of phototropies, and they regulate phototropism in plants. And one more is the cryptochromes. They found both in plants and animals. And they regulate usually circadian rhythms and might be a possible candidate to regulate magnetic reception. So as we can see, these blue light photoreceptors play a crucial role, and they do a lot of different jobs in different organisms. So, and here I would like to focus on the phototropin. That is it contains two LOF domains, LOF1 and LOF2, with then is J alpha helix and serine and threonine kinase domain here. So, and in the LOF2 domain, we have flavin mononucleotide, FMN, which is non-covalently bound to the protein. And if we have blue light illumination, it triggers a chemical reaction, and usually here we have covalent bond forming between FMN and cysteine residue at and this light-induced bond is called an adduct, and it is reversible. It breaks back in the dark. There are also interesting mutation. If we take away this cysteine and replace it, for example, with alanine, we cannot form a duct anymore, but still electron transfer happens from tryptophan, which is away about 11 angstrom from the FMN. And this makes LUF domain a perfect model system for studying how light triggers electron transfer and chemical changes, which is exactly that of what we are interested in the photo-CIDNP. So what is a photo-CIDNP? Today was already mentioned several times. It's photochemically induced dynamic nuclear polarization. And it's one of the way how we can get polarized, hyperpolarized NMR. This is the photocycle, what usually occurs in such LUF domains. Here we have flavine and tryptophan at the ground state, and after light illumination, flavine goes to the excited triplet state, and after intersystem conversion, molecular triplet, here was singlet state, and here is molecular triplet state, flavine is formed, and then we have electron transfer from tryptophan, and here spin-correlated radical pair is formed. And interconversion between triplet and singlet state is regulated by nuclear spin state. So that's why we have recombination to the ground state of certain nuclear spin states, which we can detect by NMR. In the case of NMR, instead of a typical thermal equilibrium signal, we can build up a hyperpolarized spin population and detect enhanced NMR signal. And here is one of the examples of this kind of spectrum. The first one was measured in dark condition, and the second one in light, and we see that signal enhancements quite a lot. So so here I would like to show you two examples of photo-CIDNP. Here is in liquid state, and in 2005, there was a first report observation is of 13C photo-CIDNP spectra in LUF2 domain from the plant Avena sativa. It was reconstructed by uniformly 13C enriched flavin mononucleotide. And here you see in the spectra, which is light induced, there are strong changes in the signal. And when we subtract dark spectrum from the light one, we can, this spectrum gives us information at clear view which atoms are polarized. It is possible not only in liquid state, as well photo-CIDNP polarization was observed in solid state condition in LUF1 domain. It's just one of the example. So here it's LUF1 domain from Chlamydomonas reinhardti. It's a green algae. So what is important that this protein is not carbon labeled. So, and we, the top spectrum is again dark spectrum, and the bottom one is a light spectrum. So here we see a clearly enhancement of the FMN and tryptophan signals that actually both of them, we see the enhancement from the both species which is involved in this photocycle. Actually, it opens up the door to study native system with enhanced sensitivity without the need for isotope labeling. So but here there is interesting example of the one more protein where different distance between tryptophan and flavin was investigated. This was done in work of Yonghong Ding. So, and there was a proposal that a photo-CIDNP enhancement depends on the distance between flavin and tryptophan. And we have a kind of sweet spots where we have strong hyperpolarization. It's a distance from flavin to tryptophan about 9 and 11 angstrom. And in the other case, if distance is too short or too long, there is no observable photo-kidnap detected. So, and based on everything we've seen in natural system, we decided to create our own minimalistic model Dyads inspired by enzymes. And the idea is mimic donor-acceptor architecture, which we see in flavoproteins, but in control synthetic way. So in our system, we connect a tryptophan donor and flavin acceptor using a polyproline linker, and polyproline is ideal for us because it's quite rigid and allow us to control distances precisely from short dyad to long one, and the distance variation from 5 to 30 angstrom. And this is one of the molecules that we have synthesized. It contains different lengths. We used in our work 3, 4, 6, 9, and 12 proline Dyads. And as well, it has two kind of substitution at this position, H or N-butyl. Before we moving to our results, I would like to summarize our main goal. First, we want to gain a deep understanding of the photokidnap effect, both in liquid and in solid state. And then we aim to reduce the control and optimize this effect in molecular system using synthetic models like Dyads that I just showed. And in the long term, we hope to build a platform for hyperpolarized NMR and MRI applications where photokidnap can boost sensitivity in real experiments. And here I will be focused on defining a structure photokidnap activity relationship. Now let's take a look at the polyproline helices, which we used in our system. And as we know, proline can adopt two conformations, cis and trans, and depending on that, it forms two different types of helices, polyproline helix 1 and 2. In the trans form, we get a polyproline helix 2, which is a left-handed and more extended with about 3.1 angstrom per residue. And in the cis conformations, we get pp1 helix, which is a right-handed and more compact, around 1.9 angstrom per residue. These differences are also clearly visible in circular decreasing spectrum. So we can use this spectral signature to identify which type of helix is formed in our Dyads. And actually, which helix is formed strongly depends on the solvent. In water, like it here, we mainly form pp2 helix. And for example, in alcohol like propanol, mostly formed pp1 helix. Which is cis-dominated, cis isomer dominate. And in our Dyad system, we used methanol, deuterated methanol, because it promotes more stable helix, pp2 helix formation, and as well, our Dyads, it's good for our Dyad solubility. So, and what we observed from the synthesized Dyad system, so when we have only 3 proline, this is the red line for NH Dyads and N-butyl Dyads, we have not observed this fingerprint from the pp2 helix, but with increasing proline residues, we see that the typical pattern of pp2 helix became more enhanced. So with this one, we can conclude that with more proline in our Dyad system, we build up more strongly trans conformational helix. And as well, we can detect it. We can detect it qualitatively with a noisy and a toxic NMR spectroscopy in combination. Here is the results that for different amount of prolines, we have different amount of pp2 helixes. The rest of them, they are conformers, which is containing at least one cis isomers per proline. And for all dyads, the trans pp2 form is clearly dominates, but there is still a noticeable amount of cis-containing conformers. And to complete the experiments, we run a molecular dynamic simulations. As expected, longer proline chains give the larger donor receptor distances, as it's shown here. And on the video, you can see a proline 9 dyads, which shows molecular moves and fluctuates over time. Even though the structure is mostly extended, it still has some flexibility, which can influence the kidnap intensity. This simulation helps us to understand both distance and the dynamics of the system, which are the key parameters in photokidnap. So, and one of the most interesting experiments, there is a differences between an H substitution diet and N-butyl substitute diet. With a STM microscopy, we see that an H diet forms a linear structure which is nicely aligned. There's just this alignment comes from hydrogen bonding between flavin and tryptophan groups. And the image here shows that this alignment is broken when we use N-butyl substituent diet. So actually, that's in the following slide. I will show how it influences on the kidnap effect. And as Lars already told about how liquid state kidnap effect can be measured, we use as well the same insert and CV blue laser which operates at 445 nanometers to excite flavin with power about 600 milliwatt. And we attach a light through the light fiber from the top to the liquid NMR tube. And that is actually the pulse sequence, which measure directly subtracted spectrum, dark minus light, and we don't see the thermal polarization. Here is just, I would like to explain a little bit the background of it. If we measure like it is without any light, without lasers, so what we detect looks like that. This is the dark spectrum from the difference. And if we add here light, for example, at the second scan. So then we built a non-Boltzmann distribution, and the spectrum looks like that. It's already measured like this dark minus this light in this way. So, and here is one of the great advantages of photokidnap. It is simple to generate hyperpolarization using light, and you don't need any complicated or expensive setup, just a light source, and here you go, yeah. And so, from the theory, what we have expected that our kidnap intensity will strongly depend on the exchange interaction, because exchange interaction depends on the distance between two radicals, and it decays exponentially. What we expected that in a short diet, we have quite strong exchange interaction, especially this will hinder ST0 mixing, especially at high magnetic field. But with increasing lengths between flavin and tryptophan, the exchange interaction should be smaller, and the ST0 mixing should be easier. But then at the from the other side, we face a problem with electron transfer, which should as well decay exponentially. But the recent study on the proline helixes showed that the proline helixes actually make electron transfer easier, and instead of exponential decay, the electron transfer for the proline helix way more smaller, decrease way more smaller with the distance. And here we see on this side proton kidnap spectra measured in the liquid state for the different diet, and each diet has a different length of proline linker ranging from 3 to 12 residues. As expected, some diet show strong polarization, especially those with a medium linker length 6 proline. And but interestingly, we still observe clear kidnap effect even for the longest diet with a 12 proline. Here, where the donor receptor distance is over than 30 angstrom. So, and unlike to protein, here dyads have possibilities to form as well intramolecular kidnap effect. It actually may play a role in such kind of experiments. We can measure the competition between intra and intramolecular kidnap effects. But on the all kidnap spectrum from our system, we notice that not only flavin and tryptophan are polarized, as well we observe noticeable polarization on the proline protons. Actually, this was something what we didn't expect to see, and this raises the question of whether the proline actively participates in charge transfer or interacts with a radical pair, or that in a way it's influenced spin dynamics. An alternative explanation could be that a nuclear Overhauser effect involved here, and due to special proximity of these two protons, for example, to tryptophan or flavins, they might be polarized. So here we developed a modular synthetic strategy to precisely tune donor receptor distance in flavin tryptophan dyads using rigid polyproline linkers. And we confirmed the structure of these dyads using different spectroscopy methods and MD simulations, and showed that they mostly adopt PP2 conformations. And proton photo-kidnap effect is observed in solution for all dyads, and the strong enhancement is showed for 6 proline dyads, where distance is about 18 angstrom. That's actually way more longer than in protein, where as well kidnap effect observed. And we see, I didn't mention it, that N-butyl and NH substitution shows different kidnap effect enhancement. It's most probably because this butyl chain just makes a sterical hindering for possible intermolecular electron transfer. So here I would like to thank you all our collaborators, and especially our working group, and thank you so much for the attention.
Thank you, Guzel. Thank you for the great talk. It's really amazing. It's really nice. I have questions for you, but I see that in the chat they also have questions. I have to give priority to the attendees. So yes, again, very nice. And question from Alexey Kiryutin: how long do you apply laser irradiation in your nice pulse program at slide 14 or 15? Yeah, I would like to know as well.
Thank you for the question. Actually, we test a different way how we can how long we can apply laser irradiation. The optimum was found about half of a second.
All right, thank you. Next question is from Matthias Bütikofer. Thank you for the nice talk. Did you also compare the kidnap of the linked system with that of the unlinked system?
Yeah, sure. We have done comparison for the same constructed, from this flavin with the benzene ring and this tryptophan without any proline linker. And it shows a similar pattern of photo-kidnap effect, but at this kind of system, we can't control distance between them. So, but about enhancement, as I remember, that was even more for the non-linker system.
Thanks a lot for the answer. And I see we have a hand raised from Rolf Woolens. Yes, Rolf, I think, can you, yeah, maybe you can speak and ask your question. I don't know if it, maybe you need to activate your microphone.
Can you hear me?
Yes.
Okay, good. Yeah, that's easier than typing. very nice, your lecture. Yeah, so I'm very interested about the distance effect. Yeah, also of your, yeah, your kidnap effect. Yeah, of course, an exchange integral like you show here, yeah, has only the distance in it. Yeah, but of course, a J-coupling integral also will have an orientational effect, like we know from K plus J couplings as well. Yeah, trans and cis, yeah, will have different J values. So that means also in this case, an exchange integral, yeah, will also have an orientational effect. Yeah, does that mean that in your case, since you have such nice distance dependency, yeah, is that all the bonds more or less are rapidly rotating so that the orientational effect is averaged out, or how should I interpret it?
Oh, yeah, for sure. Exchange interaction depends not only on the distance, but as well from their orientations. But unfortunately, with such type of dyad design, we can't fix some certain orientations. As you have seen from this video and MD simulations, they are quite flexible. And we measured as well angle distribution between tryptophan and flavin. They're quite broad. And right now we are working on from the dyad designs that we can just with some additional groups hinder it, this motion. That's actually the direction where we work right now.
Okay, good. Yeah, like adding like a tertiary butyl group or something like that.
Yes,
yeah, large groups.
yes.
Yeah.
Or just, for example, take away this kind of very, very long bond at this place, which actually makes system quite flexible.
Okay, thank you.
All right, thanks a lot. And I think I remember some work from University of Paris, Marie Curie in France, where they were using as well some fluorinated prolines to restrict motion or to accelerate motion. So maybe something to look into as well. All right, thanks a lot again, Guzel. And with that, we are going to welcome our last speaker, Kirill Sheberstov, who is actually in Paris, not in University of Paris, Pierre Marie Curie, but with in ENS with the CNRS. And yeah, Kirill has been working. I mean, has been cited already through the presentation, and been working in the photo-CIDNP field for a long time already in Novosibirsk, and as well in was it in Darmstadt or in Mainz, I think, where you worked a lot as well on Mainz, yes, in zero ultralow field photo-CIDNP and long-lived state. And now you also in ENS working as well on photo-CIDNP and long-lived state as well. And you are going to talk to us today about this very topic of how you hyperpolarize and how can you obtain heteronuclear long-lived state by photo-CIDNP. So yeah, really excited to hear about your talk, and Kirill, the floor is yours.
Thank you, Felix. I would like to thank you for this kind of invitation and for opportunity to present our work, which indeed will be on hyperpolarization of heteronuclear long-lived states by photo-CIDNP. So the key notion of my presentation will be span around symmetry, the notion of symmetry. For example, how symmetry can determine spin relaxation, providing a background for so-called long-lived states, which Lars Kiryutin mentioned in the beginning of this nice webinar. Then I will briefly proceed with the discussion of how breaking the symmetry can help one to perform experiments in drug screening experiments, where people look for protein-ligand interactions. And then I will discuss how one can impose symmetry on proton and carbon spin pairs, providing a way to form heteronuclear long-lived states. We will then discuss the way to hyperpolarize such long-lived states by means of photo-CIDNP at zero field, and we will compare the radical pair mechanism at high and at zero fields. And finally, I will produce with a discussion of experimental results on a model photo-CIDNP system demonstrating this kind of effects. So as the name suggests, long-lived states are special quantum states which have lifetimes substantially longer than typically NMR states have relaxation rates determined by T1. For a spin pair, for a pair of two spins 1/2, if they are equivalent, one expects to have three triplet states as eigenstates and one singlet state as an eigenstate. The crucial property of these states is that they are also eigenstate of operator representing permutation of the two spins. So the triplets are symmetric with respect to permutation, and the singlet state is antisymmetric with respect to permutation of the two spins. So it turns out that relaxation, which is driven by fluctuating terms which are symmetric with respect to spin permutation, can induce relaxation within the triplet manifold, but cannot induce the transition between states with different symmetry, providing a background for long-lived states. So this idea was introduced by a group of Malcolm Levitt about 20 years ago. Here in Paris, we are interested in one particular application of these long-lived states, which is drug screening purposes. So imagine you have a small molecule where you can create these long-lived states which have extremely small relaxation rates because these states are protected by symmetry. But once the molecule binds to a protein, there will be local fields induced at the sites of the two protons, breaking the symmetry and leading to extremely large relaxation rate of this singlet state in the bound form. So that by observing a free ligand, one may detect a significant drop in the relaxation rate of the free ligand, even if the amount or the population of this bound form is very small compared to the free concentration of the free ligand, providing a very good contrast. These ideas were introduced roughly 10 years ago in the group of Jeffrey Bodenhausen. And this is a very nice approach, and they really showed that this approach can significantly outperform existing methods to detect protein-ligand interactions with NMR. However, the drawback of this approach is that there are not so many molecules where you can create this kind of long-lived states. And as an idea to how to expand this methodology, we propose to look at spin pairs formed between protons and carbons. Of course, you can think that protons and carbons are very different species. They have very different gyromagnetic ratios and therefore Larmor frequencies. So normally, the eigenstates of such spin pair will be Zeeman states, which doesn't possess any symmetry. The situation, however, changes if you bring your sample down to zero field, where the proton and carbon can be think about as they a little bit lose their identity. They produce common triplet states and common antisymmetric singlet state. Of course, in order to achieve this, the field must be very small. So condition for this is that Larmor frequency of the proton and carbon, the difference between the Larmor frequency should be much smaller than J coupling binding these two spins. And typically this occurs below 1 microtesla for, let's say, J coupling of value of approximately 100 Hertz. All right. So this idea was actually experimentally confirmed by pioneering paper coming from a group of Alex Pines and co-workers, where they looked at formic acid labeled with carbon-13 spin, and then they detected two relaxation modes, one fast relaxation mode and one slow relaxation mode, where the fast relaxation corresponds to redistribution of populations within the triplet manifold, and the slow relaxation mode corresponding to redistribution of the populations between singlet and triplet states. This idea was further expanded and looked through by group in Novosibirsk, in particular Alexey Kiryutin, Ivan Zhukov, Alexander Yurkovska, Dmitry Butker, and Konstantin Ivanov, where they showed that it is possible to observe this kind of behavior in field cycling experiments by shuttling the sample in zero field and letting it relax, and then shuttling to high field and detecting high field spectra. It was possible to see that the components of the multiplet in the carbon spectrum exhibit bi-exponential decay, fast and slow, also corresponding to redistribution of the populations within the triplet manifold and distribution within the singlet and triplet states, which is long-lived. So this is very general approach. Whenever you have carbon and protons, you may be sure that at zero field, they will form long-lived states. The problem is, however, that this carbon-13 at natural abundance is only approximately 1%. So there is a huge penalty on the sensitivity of such experiments. And if you want to be compatible with some drug screening experiments, you must come up with some hyperpolarization technique. And in this work, I will introduce a mechanism based on photo-CIDNP. So let's consider radical pair mechanism at high field and at zero field. We will start with the high field. So normally you find a photoactive photosynthesizer molecule which absorbs a photon and then becomes a radical. And typically we work with such photosynthesizers that end up in triplet, relatively long-lived state. Then a second molecule, quencher, approaches this photosynthesizer and steals one of its electrons, generating a so-called radical pair. Also, the quencher molecule contains a nuclear spins. Let's say that it will be just for simplicity one spin, which is either in alpha or in beta state. And let's further assume that nuclear spins in beta states speed up conversion of the triplet electron state to singlet electron state, so that the in-cage recombination products will be enriched with a quencher molecule containing beta nuclei states. The escape products are also enriched in this case with the alpha state, but due to very fast relaxation induced by paramagnetic species, unpaired electron, these nuclear spin states will relax very quickly to Boltzmann 50-50% distribution. The process is cyclic, so that upon repeating continuous eliminating of the sample, one can build up the quencher in beta state. All right, so this was a radical pair mechanism at high field. What's about zero field? Well, the overall scheme is pretty similar to high field, but because at zero field there is no preferential direction, one cannot polarize magnetization, which is imbalance between alpha and beta states. So that quencher molecule must contain at least two distinct nuclei described by triplet and singlet state. And here we assume that the singlet state in the radical pair speeds up conversion from triplet to singlet state of the electron state. This process is accompanied by spin flip of the singlet to triplet for the nuclei. And upon repetition of this process, one gradually builds up imbalance between triplet and singlet states. So this is exactly what we want. One may wonder what is typical range of the magnetic fields where this mechanism is active. So to answer this, the resonance frequency of the electron should be much smaller than the hyperfine interactions of the radicals, of the electrons with nuclei, sorry. So this condition is readily met at the fields of about 50 microtesla and below. So at around Earth field, one can say that this mechanism would be operational. And if you want to have a little bit more details on this process, we have recently published a review paper on Zulf NMR in progress in NMR spectroscopy. So now I proceed with a description of a model system that we used to demonstrate these effects. The photosynthesizer molecule is tetraphenylporphyrin. The quencher molecule is benzoquinone, and we will be in particular interested in benzoquinone having carbon-13 spins in it. So we worked always with molecules at natural abundance. The experimental protocol is pretty simple and highlighted on this slide. So we start by irradiating sample at low fields, and then we bring the sample into a benchtop NMR spectrometer for the detection. Here you can see a short video demonstrating how this experiment works. So a robotic arm picks up the sample and brings it into shielded environment where illumination occurs. And then it brings the hyperpolarized sample back to an NMR spectrometer for the detection. So what do we expect for a pair of carbon-13 and proton in such kind of experiments? If we perform irradiation at ZULF, so at zero field, as I explained, we expect from radical pair mechanism to polarize imbalance between the average population of triplet and singlet state, which is then adiabatically transferred to high field for the detection. The population, in this case, the distribution of populations will be the following. The alpha-beta state will be overpopulated with respect to the other states. And for those of you who are familiar with the parahydrogen kind of experiments, this is somewhat reminiscent to Altadena type of experiments. If, on the other hand, we perform irradiation at Earth field, we still polarize singlet state, but then it's not eigenstate of the spin pair between carbon and proton at this field, because they are already weakly coupled, so that the singlet state is redistributed between alpha beta and beta alpha evenly. And then upon high field, upon the transfer of the sample into the high field, one observes this kind of initial state, which is reminiscent to Pasadena kind of experiments. All right, so now let's see what we observe in the experiments. This is a proton spectrum showing signals of porphyrin, of the solvent, and of benzoquinone. In these cases, we couldn't see any signals coming from the molecule containing carbon-13 signals because the sensitivity was not sufficient. However, when we performed irradiation at Earth's magnetic field, we suddenly saw a very strongly hyperpolarized carbon-13 satellites having very characteristic antiphase appearance, which corresponds to this Pasadena type of initial state polarization. When we performed irradiation at ZULF conditions, we see that only one satellite is polarized, but we also see carbon-13 spectra signals, and they are negatively polarized, so providing a spectroscopic signature of this population imbalance, therefore confirming that indeed at zero field we had heteronuclear long-lived states. Importantly, all this work was done on natural abundance of benzoquinone, and we see that the hyperpolarization levels are pretty high, very like reaching several hundreds, allowing to detect us carbon-13 spectra and carbon-13 satellites in proton spectra of benzoquinone at millimolar concentrations with a benchtop NMR, which is a pretty good result, I would say. Now, can we see how these long-lived states decay over the time? Can we prove that the state polarized at ZULF is indeed long-lived? In order to do this, we repeated experiments and introduced additional storage delay at low field immediately after preparation of the hyperpolarization. And then again, we transferred the sample into the benchtop NMR for the detection. And we acquired the spectra as a function of storage time. And indeed, we saw that in case where we performed this experiment at zero field, the relaxation time of the signals was approximately twice higher than compared to relaxation times observed at Earth field. So proving that this is indeed heteronuclear long-lived states hyperpolarized by photo-CIDNP at ZULF. With that, let me conclude. So we showed that heteronuclear long-lived states between carbon and proton spins can be hyperpolarized by photo-CIDNP at zero field. We observed very high polarization levels, allowing observation of carbon-13 signals at natural abundance for millimolar concentrations with benchtop NMR. And in fact, this is still ongoing project. So we believe that this is a quite general approach that might be useful for drug screening. So currently in Paris, we are reproducing this setup, and we will start soon to work on molecules which are interesting for drug screening. And in particular, we will do it in collaboration with Felix Torrens and Roland Rick. With that, I would like to thank my collaborators. So in Novosibirsk, I would like to acknowledge contribution of Ivan Zhukov, Alexey Kiryutin, and Alexandra Yurkovskaya, and of course of Konstantin Lvovich Ivanov. I would like to acknowledge my former colleagues from Mainz, Lyubov Chichkova, Inan Hu, Oleg Tretyak, Dmitry Budker, John Blanchard, and Danila Barsky. I'm currently looking for a PhD student and a postdoc interested on working on this project. So if you know some people, please let me know. And thank you for your attention. I would be very happy to answer your questions, if any. Thank you.
Thank you so much, Kirill. Amazing talk, like always. And thank you as well for making opportunity for students to join your lab and to advertise positions. I think that's really good that we can also use this type of opportunities to, advertise to a focused community that is interested in the question. And yeah, I really appreciate that. I think we have already a question in the chat. That's an acknowledgement from Ron Nauler, so discoverer of photo-CIDNP, who says, thank you to all of the presenters. Your work is breathtaking, and I can only agree. This was a really, really nice session, really diverse work, and I really enjoyed a lot. Kirill, I have a question. So you made the transfer, from the robotic arm, and you have this resting time for the LLS. Have you tried to bring the sample into the benchtop and wait to see if the LLS was confirmed as well at benchtop field?
Right. So at benchtop fields, protons and carbons are very weakly coupled, so that it's not longer eigenstates. The long-lived state cannot be observed at benchtop NMR, unfortunately, for carbons and protons. So you really, in order to impose this symmetry, you really have to go to zero field for carbon and yes, between carbon-13 and proton spin.
And a quick follow-up, because in the first talk from Alexey, we saw that there was that having LEDs could induce some magnetic field in the setup. Is it something that you observed as well? I think you have LED setup for your ZULF, right? Is it something
exactly.
that you observed as well?
Yes, exactly. We observed that the field induced by currents in LEDs goes up to 1 microtesla approximately. So, but 1 microtesla still may be considered as zero field for directly bound carbon-13 and protons. So this is less of an issue. When you work with nitrogen-15, on the other hand, you really want to go to some few nanotesla or or 10 nanotesla field to consider it to be zero field. Yes.
Okay, very interesting. And I like a lot to mention that depending on the application, you have different constraints, and you can allow yourself certain stuff. And I think that's, that's really good that we can mention that in this type of talk. So thanks a lot for bringing that. And we have a question from Alexey Kiryutin. Kirill, do you think that Kuhn being not well symmetric makes LLS not so long? Do you think that molecule with higher symmetry will make LLS longer?
Yes, I think this is maybe true because there are many protons which actually can induce dipole-dipole interaction, dipole-dipole source of relaxation on the long-lived state. So the answer is yes. If you have more isolated carbon-13 proton pair, this would be beneficial for such kind of experiments. Yes. But even regardless that we saw that the relaxation was still slower than normal T1.
Excellent. Thank you so much. I think someone raised his or her hand, but I cannot see it anymore. So if that person still want to ask the question, please raise your hand or write a comment. All right. In the meantime, I would like to thank all the speakers of today for the amazing presentation. This was really, really nice. Really glad to see that in the photo-kidnap field, we have so many different topics. Yet we have a lot of cross relationship between the different talks. We could see that a lot of stuff were intertwined, and yeah, it's really amazing to see that, and to see that this is dynamic research is going on. And probably a lot of applications are yet to come. And we have a lot strong fundamental science that is moving into more and more biological systems, and we can see a very bright future for photo-kidnap in the field. So thanks everyone. If you have questions that you would like to, that you still think about, I will take some questions until tomorrow. You can drop us an email or send us on LinkedIn at Nexema on LinkedIn some questions for the speakers. I will forward them your questions, and hopefully they will answer within the next couple of weeks, and we will send an email to everyone who attended for having the question and the recording of the talk. Again, let's thank again all the speakers, and I wish everyone a good evening, and thanks a lot for attending those talks. This is really nice to see so many people joining and seeing the interest for this topic. All right, so with that, I wish everyone a good evening, and I will end the meeting.
Bye-bye.
Bye.
Bye-bye. Bye, everyone.
Thank you.
Bye-bye. Thanks.